Hello, everyone. Welcome.
Hi.
Welcome, welcome. Happy Tuesday. If you would, go ahead and type your first name or your nickname into the chat for me. So I'll have attendance for today. Thank you very much. Very good. Looks like just about everybody's here. So we'll go ahead and get started. I hope everybody's doing well and you're settling into your schedule. I know things are a little chaotic right now on campus in terms of testing and everything. So I hope everybody's doing all right there. Obviously, we have had some increases in case numbers at UNH and at Plymouth State, and so we're trying really hard to prevent that from happening in Keene. But I know it just puts more on your plate to have to be tested anymore. And I know it's hard. I know you're all under a lot of stress. So like I said, I hope things are going well. It looks like so far everyone's doing well on their lecture quizzes, and I'm really pleased to see that. Keep up the good work. It's these lecture quizzes may not feel like a lot because they're only worth 5 points, and the lab assignments are only worth 5 points, but those points are going to add up really quickly for you. And most of my students tell me that the quizzes actually help them in the end. They help their grade, because they can be sort of a buffer against the exams sometimes that some students find, to be more difficult. So keep up the good work with those quizzes and lab questions. All right, today we're going to be talking about aseptic technique in the microbiology laboratory. But before we get started on that, I just want to take a look at what's coming up for us. So this, you should hopefully see on your screen an image of our syllabus. I'm just scrolling down in the syllabus to the schedule here. So of course, this week you'll be learning in lecture about some of the history of the microbiology science, as well as a little bit about how microscopes work. Not the physical structure of the microscope, but how it actually can create images for us and so on, and the different types of microscopy. And then there's also a lecture about the cell and cell structure this week. And again, we'll be doing aseptic technique for our lab exercise today. Just a reminder, there's no laboratory Zoom meeting on Thursday. So you'll have that time available to do some work. And then looking ahead to next week, you have your first lecture exam on Tuesday. I sent out an announcement earlier today that has just the basic information about this exam in it. How long you'll have to take it, when you can take it on Tuesday, and so on. So if you haven't seen that announcement already, go ahead and when you have a chance, go ahead and look for that. And again, it's just the basic information about the exam, what's going to be on it, and how much time you have, and so on and so on. We do meet on Tuesday next week. Even though you have the exam, you'll be taking the exam at some point during the day. We will meet for our regular lab at 4 o'clock. We'll be doing an exercise in gram staining. But make sure you've got that on your personal calendar, that exam's coming up. The most important thing I can say, which I don't believe I put in the announcement, it just slipped my mind, but the most important thing I can add to that is that you are allowed to have your lecture notes by your side while you take the exam. So the notes that you've been taking for yourself as you've been watching these video lectures, you'll be able to have with you during the lecture exam. I think that's fair. Um, just remember, you are not allowed to use any other materials. So you're not allowed to Google anything during the exam. You're not allowed to get any help from anyone during the exam, but you are allowed to use the notes that you've made. So between now and next Tuesday, over this next week, the most important thing that you'll want to do really is make sure that your lecture notes that you've taken are in good shape, nicely detailed with all of the information um, that gets stressed in the lectures. And then you'll have that available if you need it while you're taking the exam. So that's next week. Let's just quickly take a look just so we keep in practice of doing this. You should now see our homepage in Canvas, Micro for Health Professionals. I'm going to click where it says modules. And I'm going to scroll down to this week right here. So of course, here's the to-do list that you'll find in each week. There'll be a new to-do list. The slides, in case you want to make use of those. Here's that first lecture quiz, the second lecture quiz, and then the lecture quiz for the cell. And then this is the lab questions that you'll be doing this week about today's lab. And then if you scroll down to next week, you'll see, again, the list of things that have to be done next week. And here's the lecture exam. I've got a little note on you for you. Don't click on it until you're ready to start, because you're only allowed to take the exam once. And Canvas is a little funny about it. Sometimes when students click, thinking that they just want to see the instructions, Canvas will count that as your attempt. So don't click on that link here until you're ready to start the exam. I don't want you to worry about the exam. If you've been watching the lectures and taking good notes for yourself, you're going to be just fine. Most of the students who take this exam finish it in about 30 to 60 minutes, and you have a full 90 minutes to finish it. So I don't think you have any reason to be worried about this exam next week. Okay. Does anybody have any questions about any of the lecture material you've been watching, or questions about any of the quizzes, or anything at all that you'd like to mention or talk about? We can always do that during these lab periods if you have any questions. It's always a good idea while you're watching lecture videos to keep some blank paper near you that you can jot down if there's anything that doesn't make sense to you and you'd like to review it. Like I said, the quizzes that I've seen so far are great. I mean, 4s and 5s across the board. So it seems like you're doing well with the lecture, which is wonderful. Keep it up. All right. Well, if there's no questions, I'm going to get started then by pulling up our lab slides for the day. Remember, you can, our Zoom lab meetings are recorded. So if you have, while you're taking notes today, if you miss anything or you want to go back and watch something again that we say today during our meeting, you'll be able to find a video recording of this meeting on our playlist later on today. All right. So you'll always be able to go back if you miss something. All right, so I'm going to pull up our slides for today about aseptic technique. The objectives that we're trying to meet today in this lab exercise are first to define this. What are we talking about when we talk about aseptic technique? We're going to talk about why these techniques are used in a microbiology lab. What is it about these techniques that makes them so useful to us? And then we will talk about how to implement these techniques. And I'm going to go through some video with you to show you how to implement some of these techniques that we refer to all together as aseptic technique. So when someone talks about using aseptic technique, they're really talking about using a whole variety of techniques. So the definition for aseptic technique is all of the methods that we use in the laboratory in order to prevent contamination. As we talked about, it's all about preventing contamination. We don't want to contaminate the cultures that we're working with or the diagnostic samples we're working with, and we don't want to contaminate ourselves. That's really our very first goal. Always, always, always has to be in the back of our minds that we don't want to contaminate the culture with any organisms that are on us or in the environment. And we don't want to contaminate ourselves with anything that might be in the culture. It's important to remember, and you'll hear me say this a lot this semester, most of the microbes in this world are not harmful to us. Most of them are either completely harmless or even helpful to us, the ones that live on us and in us. But there are pathogens. There are a number of particularly bacteria and viruses that are pathogens. So whenever we work in the microbiology laboratory, we treat every culture container as though it contains pathogens. If we do that, we will get in the habit of handling those culture containers very safely so that should someday there be a pathogen in one of those containers, we'll be safe. We treat every culture that we handle as though it contains pathogens. Now, when we grow when we grow microbes purposefully in the laboratory, and of course we do that all the time in microbiology labs, we purposefully grow these collections of microbes in these media, as we call them. The culture is the container that has both media in it and microbial cells, whatever they happen to be. So those two things combined together are what we call a culture. And of course, we put the culture into the incubator most of the time because it needs darkness and it needs warmth in order to grow most efficiently. Sometimes, though, we do incubate things in room air. There are some microbes that would prefer to be in room air. So we don't always put cultures into the incubator, but most of the time we do. This culture media, this material that we're using to grow the microbial cells in, it can be solid, it can be semi-solid, and it can be a liquid. So all three forms are used. And these media contain the nutrients and the other molecular materials that these cells need in order to grow. Because again, we want them to grow. We want them to divide and make new cells. That's the whole reason we culture microbes. We say that we inoculate our culture media with these microbes. So for example, if I have some solid culture media, if I have some solid agar, as we call it, in a petri dish, I'm going to inoculate that dish by spreading some kind of a microbial organism over the surface of the agar. That's called inoculation. I also might handle a liquid media, and of course we call those broth. And I can inoculate the liquid by putting some cells into it. This inoculated media then is what gets incubated. And again, a lot of the time we're incubating inside that little chamber called an incubator. And we can control the temperature and the humidity and the amount of gases inside that incubator in modern incubators. There are some organisms, for example, that like a little bit more carbon dioxide than what is in room air. So modern incubators, we can set to all kinds of different parameters in order to give those microbes the conditions that they really like, so they'll grow for us. And again, incubators are nice and dark too, which most microbes prefer. But there are some microbes that you can incubate in room air. So we literally just either put them into an incubator that's turned off. That's a nice way to do that. Or you can leave them on a countertop as long as they're not in direct sunlight. Remember, sun is a great disinfectant. So you don't want to leave your culture containers in the sun. Now, it's important to remember, going back to our goal about not contaminating things, these media that we use to grow microbes in culture, these are designed for microbes to grow. We're purposefully putting in all these nutrients and things into these media. So, yes, the organism that we're trying to grow is going to grow in there. But if a contaminant happens to get in there, if another microbe happens to get into the container, that's going to grow as well. So remember, these culture containers that have media in them, they are perfect for microbes. And so we have to work very hard to keep contaminant organisms out of those containers. We always have to handle culture media very carefully so that we prevent that kind of contamination. Now, there's a lot of information on this slide, I know, but I put together basically a list of the most general rules that we try to remember whenever we're creating these cultures or taking the culture and doing something with it. Perhaps we're going to remove some cells and start a new culture, or maybe we're going to take some cells out and do some testing. Anytime we're handling it, we have sort of a short list of general rules that we try to follow. Number one, we never want to leave a culture container open any longer than is absolutely necessary. So whether it's an agar plate, which again is a petri dish that has agar in it, whether it's an agar plate or a broth test tube or any kind of culture media container, we don't want to leave it open any longer than we absolutely have to. Because anytime that it's open, there's a chance that contaminating microbes that are in the air might get into that container. Number two, we're always going to assume that surfaces are contaminated. So that's even if you just started working and you just disinfected your workplace with alcohol. Even if you just did it and you know that it's sparkling clean, you have to assume that that surface is contaminated. Because if you do that, it will tell you never to put anything that's sterile down onto that surface. So anything that we handle that needs to stay sterile, we keep in our hands. We don't put it down on the desktop. The third thing on the list is for us to always remember that microbes fall down out of the air. They don't jump up or fly up. So in other words, if I simply open an agar plate that has cells growing on it, those cells cannot jump up off that plate and contaminate me. The only way I can contaminate myself is by touching the cells. If I open up that plate though, and I leave it open to the air for too long, organisms that are in the air can fall onto that plate and contaminate it. So again, we don't want to leave it open any longer than necessary because we want to minimize the chances that a microbe is going to fall out of the air and land on that plate and start to grow. Nearly all of the contamination that we get in our cultures in microbiology labs is in the form of dust particles that are being carried in the air currents in the room. The dust particles, that you and I, we just can't see them. And those dust particles are literally carrying these bacterial cells and viral particles and fungal cells and things like that. And they're traveling around in this air because in most laboratories, at least, the air has circulation. There's a ventilation system in the room. So the air is moving. And unless we're under one of those specialized biological hoods, there's a risk that those contaminating cells and particles that are on those dust specks in the air are going to land in our cultures and cause us problems. So we have to be very careful about that. Now, we said a little bit about how we handle things, how we handle trash and things like that when we were talking about biosafety. There's not really a better place for us to discuss this than along with this topic today. So I've put in some slides here, again, that we can think about as part of our aseptic technique, part of preventing contamination. And that's how we dispose of things that we're done with in the laboratory. Now, some of these things are not directly related to aseptic technique, but again, this is a good time for us to talk about all of our waste disposal guidelines. Remember, we said the stains that we use in the laboratory. Sorry about that. That's my dog giving her best howl. The stains that we use in the laboratory are all considered hazardous. None of them go down the drain. So of course, we have to have hazardous waste containers in the laboratory for those stains and for any other chemicals that we might use that are considered hazardous. And again, we would have to look at the label on the container to know if it's considered hazardous. Of course, we also use regular trash cans in the lab, and there are a lot of the materials that we handle just get thrown away like regular trash. The gloves that we wear get thrown into regular trash. Paper towels that we might use to clean up spills, those Kim wipes that we might use, lens paper that we use on the microscope. Those kinds of things get used all the time in the micro lab, and they don't need any special handling. They just go in the regular trash. Sharps containers are very commonly used because we do use some sharp instruments that are single-use items and get thrown away. If you know about sharps containers because of your workplace, if you've ever worked in a doctor's office, for example, or assisted living facility, or if you've ever worked in a hospital space, you know that that sharps container, that red box that is used to throw away sharp things safely so that the people who handle our trash don't get stuck with those sharps. Now I've got a star next to the word sharps container here because in a micro lab, we actually use two different types of containers. I'm going to say a few more things about this in just a minute, but we actually label our two sharps containers in the micro lab differently. One of them is going to be a regular old contaminated sharps container. So it's going to have a biohazard symbol on it. And again, I'll show you that in just a minute. These are the kinds of boxes that you would see in a doctor's office. They're a red box with a plastic top, and they have a biohazard symbol on them. And they are for sharp things that have come into contact with something biological that might be able to contaminate us. So, any sharp object in the micro lab that has come in direct contact with our bacterial cultures, those things have to go into this contaminated sharps container. But I am going to talk just a little bit about this other sharps container because there is a type of sharps container that you'll find often in the micro lab that is not for contaminated things. And again, I'll talk about that in just a minute. Remember that because we use a lot of glass in the micro lab, we use all those microscope slides. We always have a glass waste container. We'll take a look at that. And of course, we always have to have access to autoclave bags. And I'm going to show you what one of these looks like in case you've never seen it. These are special plastic bags. Again, they're going to have a biohazard symbol on them. And that's an indication to us that the things that are inside that bag have touched either body fluids or microbial cultures, and so have the potential to contaminate us. And of course, anything that goes into an autoclave bag is going to get put in an autoclave and sterilized before it gets either disposed of or reused. Some of the things we handle are single-use items, so they're just going to get autoclaved and then disposed of. And other things that we use are reusable. Think about glass test tubes, for example. We will clean glass test tubes and then autoclave them to sterilize them so we can use them again. All right. So let's talk about these aseptic techniques, all of the different methods that we use every day in the lab that are designed to prevent contamination. We've actually talked about some of these aseptic techniques so far. For example, decontaminating or disinfecting your workplace is considered part of aseptic technique. And remember, we choose either alcohol or bleach typically to do that. Either 60 to 90% alcohol, and most of the labs I've been in use 70%, or we can choose 10% bleach. So we're always going to clean off our workplace. We clean it off before we get started, and then we clean it again when we're finished before we leave for the day. We talked about properly organizing our personal materials and the equipment that we use during the day. We need a nice, orderly workplace. We talked about how you have to sterilize the inoculating loop, that little tool that we use over and over again to transfer microbes from one culture container to another, or from the culture container to a slide. Anytime we have to move microbial cells, we use these loops to do it, and they're going to have to be sterilized. Now, we sterilize our loops, at least our metal loops, because there are some other types that we'll learn about later. Our metal inoculating loops have to be sterilized before we use them, right? If I'm going to put my loop into a pure culture of bacterial cells, I have to make sure it's sterilized before I do that, or else I might accidentally contaminate my culture. So we're going to sterilize that loop before we use it. Then we're going to use it for some purpose, and then we're going to sterilize it again. If we touch it to any kind of microbial culture, we have to sterilize it before and after we use it. So those poor metal loops get sterilized all the time. They get sterilized again and again and again during the day. It's the wire portion of these loops, and you'll see this later in the video we're going to watch. It's the wire portion of these tools that are going to get sterilized. And in a lot of microbiology labs, we're going to use the incinerator instead of an open flame. There are some labs that are still using open flame. It's not that uncommon. And if you're using an open flame, you just want to make sure you get that wire red hot in the flame. That will sterilize it. But a lot of the newer laboratory spaces are using incinerators. And these are those devices that have a ceramic tube in them that gets very, very hot. We put the wire into the tube and we leave it again until it gets red hot. You want to see that red color in the wire because that tells you that all the cells that might be on that loop and that wire are now gone. We've burned them up and we've sterilized the wire. It's important to remember, though, this is a common mistake for us when we're first learning. If I want to take some cells out of a culture and I'm sterilizing my loop before I do that, I have to remember to let my loop cool off a little bit before I put it into the culture container. If you put a hot loop into say a broth tube, you're going to get a loud sizzle sound because you've just killed a bunch of microbial cells. It's not the end of the world. You haven't killed the entire culture tube or anything, but it's just not good practice. So what we do is we take our loop out of the incinerator and we let it cool off for about 20 seconds before we put it into any containers that have cells in them. So again, we've talked about some aseptic technique already. Today, we're going to learn some new aseptic technique, some of the methods that we use, again, to prevent contamination while we're working. Today, we're going to talk about how we flame a glass test tube and why we do that. We're going to talk about how we transfer bacterial cells from a broth culture tube into a brand new sterile culture tube. In other words, let's say I have a culture of bacterial cells growing in the incubator, and I want to keep those cells alive. After a couple of days, I'm going to have to give some fresh culture media to these cells. So I take a few cells from the old culture and I transfer them into a brand new tube of culture media with all kinds of nutrients for them so they can start and continue to grow. We're going to talk about how we transfer bacterial cells from a broth tube onto an agar plate. Remember, that's solid media. We'll talk about how we handle these agar plates aseptically. And we'll look at a couple other tools that we use beyond the inoculating loop, as well as that waste disposal bag called an autoclave bag that I mentioned a few minutes ago. But before we jump into the demonstration, I do want to talk about these sharps containers. These two different sharps containers may be encountered in other labs, but they're commonly encountered in the microbiology lab. Remember, this red box, this commercially available red box that you buy from laboratory suppliers, this is what's considered a sharps container, or more correctly, a contaminated sharps container. If you look at the label, and I know it's hard to see, there's a biohazard symbol right there. There's a universal symbol for biohazard on this box. And what that tells people who touch this box is that the sharp things that are inside of here have touched potentially pathogenic materials. So materials that could potentially contaminate me. Things like body fluids, blood, urine, anything like that. Or microbial cultures. So this box contains all those sharp things that have been contaminated. When the box gets filled up, it gets autoclaved. So all of the danger gets removed from those items, and then it gets disposed of as just sharp waste. The sharps containers that you find in micro labs that look like these red boxes, the same ones that you will find in hospitals, in doctor's offices, in ambulances. Those are all sharps containers that are designed for contaminated materials. Once they're full, they get treated like biohazardous waste. They are treated as though they can harm us by contaminating us with pathogens. And that involves autoclaving them before they're disposed of. So in micro labs and probably other labs too, there is a second sharps container that you should know about. There's another type of sharps container that is used for sharp objects that have not come in contact with anything potentially hazardous. So let's say I use a razor blade to scrape some tape off of a container, and I'm all done with it, and the blade's kind of dull and I don't need it anymore. I'm not going to throw that razor blade into the regular trash because it could poke through the bag and harm the person who empties the trash. So instead, I need a sharps container for it. But it hasn't touched anything contaminated. It hasn't touched any culture material. It hasn't touched any body fluid. So I'm going to put it into the non-contaminated sharps container. Now, you might ask yourself, why not just put it in the regular sharps container? Why not just put it in the red box? Well, the answer is that it costs money. It costs money to get rid of those red biohazard sharps containers. So we keep 2 sharps containers, and we try to only put things that have actually touched the culture material into the contaminated sharps. We try to keep our other sharp things that we're throwing away in a non-contaminated sharps container. These things are going to get thrown out just like any other trash. They're going to go to the landfill, but we want to keep them contained because we don't want anybody to cut themselves. The danger here is not getting contaminated with a pathogen. The danger is that something is in here that's sharp and it might cut you. In this container, there are 2 dangers. The danger is something is sharp, and that sharp thing has touched culture or touched body fluid, and it could potentially harm us. These boxes have to be autoclaved, and that's expensive. These boxes don't, or I should say these containers don't. So it's really just a money thing. We have to save money. Again, these commercial boxes, you've got to autoclave these, and that adds a cost. So when we use a non-contaminated sharps container, those aren't containers that you buy commercially. Most of the time, those are just old containers that somebody has saved and brought in from home. For example, you might see an old laundry detergent container used as a sharps container. You might see an old coffee container, an old coffee can or something. The only regulation, and of course, there's all kinds of regulations about lab safety. The only regulation about non-contaminated sharp materials in the lab is that we have to put them into a container. The container has to be labeled as sharps, and the plastic or whatever the container is made out of, it has to be opaque. In other words, you can't see through it. You don't use clear material for sharps containers. So again, there's lots of plastic containers that people generate at home, and they can sort of repurpose them in the laboratory as sharps containers. When these get full, they don't have to be disposed of in any particular way. Somebody just needs to take them to the recycle center so they can be disposed of properly and safely. So you would never use an empty milk jug as a sharps container because, again, these are see-through plastic, right? And the regulation tells us that we don't want see-through plastic. And of course, you would never use a soft material. It has to have a hard a hard material in it so the sharp things don't poke through. So all you have to do is put a little hole in the lid. Put a little hole in the lid and people can dump their disposed sharp things in here. Just a little something to know about in terms of different sharps containers. I mentioned a few minutes ago that we dispose of glass in a particular way. This is a glass disposal container. These are also commercially available. You buy these from laboratory supply. This box is probably about 3 feet tall, maybe a foot wide. It's made out of really heavy cardboard and it has a hole in the top. So what drops down in here are the glass things that we're disposing of, we're throwing away, that have not come in contact with living cells. Now that's an important distinction. We do put our microscope slides in here. Even the ones that have cells on them, microbial cells that we have stained and examined. Now, the reason we're allowed to do that, the reason we can put slides in here that have bacteria on them is because when we stain these slides in the laboratory, the cells die. So those cells are no longer a danger to anybody once they're put on a slide and stained. When we're done, we just throw them right in here. Remember, glass microscope slides are generally single-use items. There are exceptions, but most of them are designed to be used once and thrown away. And because glass can cut people, we want to dispose of them safely. All right. Any questions about any of the things I've mentioned? Y'all have probably seen on campus in certain labs these two different kinds of sharps containers. Some labs don't have any contaminated sharps containers in them because they don't ever handle anything potentially pathogenic, right? So if, for example, in a chemistry lab, the only sharps container you're going to find in there is, the ones that are homemade. But if you go into a biology lab or you go into a microbiology lab or a research lab that handles living cells, you should see at least the contaminated sharps container in there. And of course, the contaminated sharps is the one you see in any healthcare setting. Any questions at all about what I've said so far? What we're going to do now is we're going to watch a video. It's 25 minutes long, and it's a demonstration of these new techniques that we haven't talked about yet. And it's primarily moving cells from place to place, and some of the other tools that we'll use this semester. All right, let's take a look. So on your screen, you should see an image of an incinerator sitting on a countertop. There's a piece of paper towel, and there's an inoculating loop sitting down here. So I told you a few minutes ago that we always assume that surfaces are contaminated in the microlab. So if I look at this picture and I see this loop, I told you, you can't put the loop down. You can't put a sterile loop down, because if you do, you have to assume that it's now contaminated. But that's okay. If I want to use this loop, I just put it back in the incinerator and sterilize it again and then use it. You would never put a dirty loop down onto your desktop. But if you know your loop is clean because you just sterilized it, it's okay to lay it on the desktop as long as you remember to sterilize it again before you use it. All right, let's get this started. When we're working in the microbiology lab, we are often moving bacterial cells from one place to another, from one media to another. And there are different... Here, hold on. I'm going to try to make it bigger for us here, if I There we go.
When we're working in the microbiology lab, we are often moving bacterial cells from one place to another, from one media to another. And there are different reasons that we do this, but we always have to use our very best aseptic technique while we're transferring cells so that we don't cause contamination to occur. Now, there are lots of reasons why we would take cells out of one culture container and move them to another. One of the most common reasons we do this is because cultures get old. Remember, when we put cells into, for example, a broth culture, there's a certain limited number of nutrient molecules in there. And as the bacterial cells start to grow and divide, they're going to use up those available nutrients. They're also going to be excreting waste materials into that culture tube. And that's going to be dangerous to the survival of the bacterial cells. So from time to time, if we want to keep a culture going, we have to take some cells out of the old culture and move them into fresh media so that they can begin to grow and divide, again, with plenty of nutrients and plenty of space. We call that process subculturing. When you take cells out of an existing culture and you move them into fresh media, we call that subculturing. Now, as you've seen, there are really two primary types of culture containers that we encounter most often in the laboratory. There are test tubes, and those can have liquid media in them. They can have a semi-solid media in them. They can even have solid agar in them. The other container we see is the Petri dish, the plastic single-use lidded dish. We often refer to these as plates. And those agar plates have a thin layer of solid media on the bottom. So those are the two containers that we encounter the most, and those are the two containers that we move cells between most often. So what we'll look at in this video is how to do all of that moving using our very best aseptic technique. Set up in the workspace here, there's an incinerator that's been plugged in and is nice and hot. And there's an inoculating loop. The inoculating loop must be sterile because we would never lay a contaminated inoculating loop on the desktop like this. The only other piece of equipment that I'm going to need to do this demonstration is a Bunsen burner. I don't have access to a Bunsen burner today, so I'm going to use something as a substitute, just for demonstration purposes. And that something is a bottle of hot sauce. That seems appropriate, a good substitute for a Bunsen burner. Now, the next thing I need is a broth tube. And I want you to notice that it's a glass tube. We've talked about how to open up any test tube with culture in it by holding it at an angle and by holding on to the top of the tube in our fingers. What we need to remember is that the inside of a sterile tube of uninoculated broth is still sterile until we place something into it. The outside of the tube, though, once you have touched it, it is no longer sterile. Now, when we're working with a glass test tube, we can take an extra step to protect the sterility inside that nutrient broth tube. And that's to flame the mouth of the test tube when we open it, before we handle it, before we put culture material in, and also flame it again before we close it. Now I'm going to show you what I mean by flame it. This is something we can do using aseptic technique to add an extra layer of protection against contamination. People don't always flame glass test tubes. Sometimes it's just too time consuming of a procedure, but I want to show it to you because it is, again, an extra layer of protection against accidental contamination. You would never do this with a plastic test tube, though, of course, because you would melt the plastic. So the test tube that I'm holding in my hand has a liquid broth in it, a nutrient media, and bacterial cells growing in it. And I'm going to take a sample of those cells out of this tube, and I'm going to flame the mouth of the tube before and after I remove those cells. So you'll notice that I'm going to take the cap off. I'm going to hold the tube at an angle, just like always. But here's the extra step. I'm going to pass the neck of that tube, the mouth of that tube, through that flame. Let's take a look at that again. Remember, the bottle of hot sauce is my Bunsen burner, and we're pretending there's a flame coming out of that black cap. So I'm going to take the neck, the open part, the mouth, if you will, of that test tube, and I'm simply going to pass it through the flame. Now, what does that achieve for me? The opening to the test tube is the place that's most vulnerable to a contaminating microbe falling in. My hands are up near the mouth of that tube when I'm taking the cap off. And of course, I'm also a source of contaminating microbes, even with gloves on. So, by flaming that opening to the tube, what I'm doing is I'm applying heat directly to the glass. So if there's any microbes sort of right on the lip of that opening, I'm killing those. I'm also briefly warming up the air that's in the mouth of that tube. And by doing that, I actually create a little current, a little swirling current of hot air. And just for a moment or two, that's going to protect the tube. It's going to prevent anything from falling down out of the air and going into the tube. So I've created a few moments of protection from accidental contamination. Now, after I'm finished doing whatever it is I'm going to do with the tube, I'm going to pass it through again before I put the cap back on. Again, if I have accidentally introduced any microbes during my procedure, I just killed them. And I've created a little current of air right at the end, just in case there were any microbes hovering over that area, that opening. I've killed them with that heat. It's a very brief pass through the flame. It's going to kill cells that might be on the lip, the outer lip of that tube that might be a source of contamination. And it's going to create a current of hot air just in the mouth of the tube, just for a couple of seconds, that will keep that entryway free of contaminating microbes. Now we're going to watch a transfer of cells from one tube to another tube. This tube has my bacterial culture in it, okay? And this tube has clean nutrient broth in it. Notice that I put the clean broth tube up higher in my hand. I'm going to widen our view here a little bit. Now, the other thing I'm going to need is a Bunsen burner, and of course I don't have one, so I'm going to substitute in my bottle of hot sauce. Okay, now, first thing I'm going to do is sterilize my inoculating loop. That needs to be into the incinerator until the wire is red hot for purposes of the demonstration, I'm going to shorten that up a little bit. We'll pretend that we're red hot. Now we have to hold it until it cools off. Remember, approximately 20 seconds. You can count in your head. Now it's important that you get that loop comfortable in your hand for the next part. I've got my tubes at an angle. I'm going to take off both caps. And the reason I do that is because I'm going to be flaming. So I'm going to flame the culture tube first. Now I'm going to take out a sample from the culture tube. I'm going to flame it again. And now I'm ready to close the culture tube. Take one of your caps and put it on. Flame the clean broth tube. Place your sample into your plain broth tube. Swirl it around a little just to make sure it all gets in there. Flame it again. Close it with a cap. Remember, your loop is dirty right here, so don't forget to put it back into the incinerator to sterilize it once again. If you're working in a laboratory that doesn't use open flame, you can still do a successful broth-to-broth transfer using your best aseptic technique. You're just going to want to remember to hold both tubes at an angle while they're open. You're going to want to avoid touching the neck or the lip of the tube as much as possible. And you're going to want to close those tubes just as soon as possible to help prevent any accidental contamination. Now let's take a look at the next kind of transfer, which is from a broth tube to an agar plate. Remember, we use plastic single-use Petri dishes in the microbiology lab, and we can pour agar into those dishes and allow it to solidify, giving us a solid media to work with. We refer to those as agar dishes or agar plates. The agar, the solid material, has nutrients added into it so that the bacteria can grow. So this is an agar plate. This happens to have nutrient agar on it. You can just see it. It's a thin sort of beige film on the bottom of the plate. Now I'm taking the lid off just so you can see the inside of the plate. There's just this beigeish sort of sheen to that media. Very, very thin layer of media in here. It feels a lot like gelatin, and it's easy to push your loop into that gelatin. So you have to be careful when you're handling it. Oh, I've mentioned before that we handle agar plates upside down. We put agar plates into the incubator to allow bacterial cells to grow upside down. In other words, we want the agar, that solid media, to be at the very top once it's upside down. And what this does is it allows any condensation that might develop inside that nice warm incubator. It allows it to drip off of the surface of the agar and onto the lid of the plate. This is helpful when we're trying to grow bacteria because that condensation might interfere with the growth of bacterial cells on the agar surface. So we handle them upside down, we open and close them upside down, and we place them into the incubator upside down. I'm going to move my incinerator into place here as I get going. My inoculating loop, my tube full of bacterial culture. Now, this inoculating loop is clean, it's sterile, and I want you to notice that I'm going to start bending this wire. The wire is wire for a reason. When we're inoculating things, especially the surfaces of plates, we can bend this wire to make it easier to spread the cells out. Now you can bend it as far as you want or as little as you want. It really depends on the person handling the loop. But once you bend it into place, you can still get it into the incinerator bent because it's very flexible. It's a wire. So you would heat up the wire just as you always would. See, the bend is still in it. You're going to let it cool. If you have it bent as severely as I do here, once you open up your culture, which I'm doing here, you're just going to want to be very careful as you introduce the loop. Again, you don't want to touch the neck of the tube. Take out some of your culture material, close your culture. Now you're ready to go. So I'm going to open this dish. Again, it's upside down. And I'm going to keep the plate at an angle so that I can put these cells on the surface. I'm going to place the loop on the surface, and I'm going to make a nice zigzaggy motion all the way across. I'm going to take my loop, remember, incinerate, sterilize, get rid of those cells. So essentially what I've done is I took a bunch of cells out of the culture tube, the broth, and I placed them onto the first place that I hit on that auger. I put a large number of cells, a large amount of cells onto the surface. And then by sliding my loop across the surface, back and forth and back and forth, what I'm doing is I'm spreading those cells across the surface of the plate. So that I'm giving them room to grow on that nice nutrient-rich solid media. Let's take a look at that again. Plate comes up, loop touches the media, and then without lifting it, I'm going to just streak it out, streaking it back and forth in a zigzag motion, spreading out those cells. As we go through the summer and we look at other techniques that we use in the microbiology laboratory, we'll also look at different ways that we can streak an agar plate, depending on what we want to achieve in terms of the growth of the cells. Now, anytime you open a container that has growth media in it, nutrient media or any other kind of media, remember what we said. You want to keep that container open for as short amount of time as possible. You want to quickly get it inoculated with cells, and then you want to close it again because you want to prevent contamination. Now, different people use different techniques to achieve this. You can open and close an agar plate a couple of different ways using aseptic. Here's an agar plate that's being held right side up. And in this technique, we're going to use the lid to keep the plate partially covered. You see that? I can reach in between with my loop and do my streaking pattern, close it, and then put it upside down. Let's look at that again. I'm going to keep the plate right side up, use the lid as partial protection from microbes falling onto it, streak it, close it, back upside down. Now let's take a look at a couple of ways that you don't want to handle your agar dish. First of all, you never want to just open up an agar plate and hold it straight up to the air like this. You're just asking for contamination to fall onto that plate. So don't do that. You also don't want to do the other extreme, which is keeping it completely upside down and trying to inoculate it. You will push your loop into that agar if you do that. What you want to do is open up the plate and hold it at an angle like this. You can get access to the surface. You can get your streaking done. Get the plate closed. You're good to go. The last thing I want to talk about today isn't directly related to aseptic technique, but it's the tools that we have available in addition to what we've talked about already. These are single-use, disposable inoculating loops. They come in a sterile package of about 50. When you open up these packages and you pull out the loop, you can see the handle end is what's going to come out first. So you widen up the mouth of the bag and you carefully pull this out, and you've got a sterile loop at the end. Now, some people don't like these single-use inoculating loops. They are not nearly as flexible as a wire loop. And obviously, they're a single-use plastic item. So you can imagine that when they're being used in a busy microbiology lab, hundreds of them are being used every day and discarded into the landfill. So some people are opposed to their use. It's a problem in all of laboratory work that we use a lot of plastic. We use a lot of single-use plastic, disposable materials. And hopefully, at some point, we will get away from that. But this is a tool that's available to us. And some people really like these because it speeds up their work. This loop allows you to do a single transfer of material and then just toss it into the autoclave bag. You don't need to take the time to put your loop, your metal loop, into the incinerator, wait for it to get hot, then take it out, wait for it to cool. Instead, you just grab a brand new disposable loop. So again, it depends on the lab you're working in, whether or not you'll be using reusable metal inoculating loops or these single-use items. Well, the next item that we can look at is the disposable cotton swab. Those are very useful in the microbiology lab. They come in these sterile packages. There's usually 2 swabs in the package. You can only open these packages at one end. And again, just like with the loops, that allows you to open the package at the end opposite to the swab. So if you carefully manipulate the package here so that the opening is a little bit wider, you can pull the swab out carefully and you've got a nice sterile surface. So you can take culture material up with a cotton swab and use it the way you would use an inoculating loop. When you're done, you can simply toss that swab in the trash. Remember, transfer pipettes also come in these single-use sterile packages. When you open these up, you're going to first see the bulb end. And again, you want to open these carefully and pull them out carefully, but now the end that will touch your liquid broth is sterile. You can pull up a larger volume with these than you could with an inoculating loop, which makes them very nice. Very inexpensive, very widely available, very widely used, these transfer pipettes. Next on the list of tools is one of my favorites. This is called a hockey stick. Now you can buy a reusable glass hockey stick, but this is the disposable variety. Now watch what this stick is going to allow me to do. We've looked at how we streak cells across a petri dish, an agar dish. But sometimes we want to make a smooth coating of cells over the surface of that dish, and that's what the hockey stick does. See, you just swirl it around like the hands of a clock face, and you will spread your cells all over the surface of that plate, nice and evenly. One of my favorite tools in microbiology. At the end of the day, when we've finished our work, it's critical that we know where to dispose of our materials. And of course, when it comes to the materials that touch culture, we need an autoclave bag. This red bag has a biohazard symbol on it, and it clearly says on it, it's an autoclave bag. So this bag is where we put used culture-related materials, the things that have touched living bacterial culture or microbial culture. our agar plates, our cotton swabs that we might have dipped into culture, our disposable transfer pipettes that may have drawn up culture, um, the cotton swabs, the hockey sticks, all of these things that have touched living cells that we cannot put in the incinerator because they're disposable. We're going to put in the bag. We're going to wrap that bag up with a tie, and that bag is going to go into the autoclave. Now, we haven't talked about the autoclave yet. We haven't talked about the process of autoclaving, but for now, understand that in the autoclave, a very high temperature is reached and a very high pressure is reached. And any cell and any spore that's inside that autoclave is going to be destroyed at that high temperature and pressure combination. Once the material comes out of the autoclave, it is ready to be thrown into the regular trash.
Okay. So some demonstration there for some of our most commonly used aseptic technique, the transferring of cells from place to place, and how we would inoculate, broth, or how we would inoculate an agar plate. Those are the most common things that we're doing in the lab every day. Does anybody have any questions about anything you saw in that video? You'll notice that I talked about all the things we're going to learn this summer. I made this video when I taught the class over the summer remotely. So it's not summer right now, unfortunately. We're all looking forward to summer, but yeah, that's why I said that during the video. Anybody have any questions about what you saw? You know, some of these techniques, when you see them, they look a little awkward. They look hard. Like when you see somebody holding two glass tubes in the same hand. And it just seems like it would be really hard and really awkward. But these things, once you start doing them in the lab, you pick them up very quickly. I promise. We don't have a chance to practice them this semester because we're remote this semester. But these things they're quick to learn. People who work with microbiology and do it every day, either do it as their job or, do it, for research or something. They get very good at these techniques very quickly, and they get very fast very quickly. So I know sometimes they look a little hard, but honest to goodness, they're not. Once you have a chance to handle them and try them. All right. Any questions? All right, very good. I'm just going to go back in here. We're going to just take a peek at a couple more slides here to finish up today. I just want to remind us that we're not going to meet on Thursday afternoon. So you have that time to do whatever work you need to get done. Watch a lecture or take a quiz or work on your lab questions, because you can now do your lab questions that are due on Sunday. You now have enough information to complete those. Remember, moving into next week, you have that lecture exam coming up. So make sure that all of your notes are in good shape before Tuesday. Again, I sent out an announcement earlier today that tells you all the really specific details about the exam. We do meet on Tuesday next week at 4:00. So you have to take the exam, and you also have your regular Zoom meeting. And during that Zoom meeting, we're going to be talking about a differential staining technique called gram staining. And that'll be next week. So if there aren't any questions, that's all I have for you today. Of course, as always, if any questions come up, just message me through Canvas. Okay. And keep up the good work. You guys are doing great. All right. I'm going to let you go. Have a good evening. And
Thank you.
we'll see you next time.
Thank you.
You're very
Thank
welcome.
you.